Hello students. So in the last lecture we have discussed about the life cycle of Riccia. So today we will discuss about the fertilization in Riccia. It is a part of the life cycles only. So mostly take place fertilization of Riccia take place in the water. So this water acts as a medium of swimming of the anthrozoid. So water is also required for the rupture of antheridium, liberation of anthrozoid and opening of the archegonial neck. So water is retained in the dorsal furrow of the thallus after rain or heavy dew. The anthrozoid is a thin film, or we can say water are attached chemotically by the mucilaginous substance oozed out by the open neck of the archegonium. The mucilagus contains chemical substance. They include soluble protein and organic salts, particularly those of potassium. So the anthrozoids swim and travel through the neck down towards the egg. A single anthrozoid fuses with the egg and forms a zygote. So the zygote is the first diploid cell which forms a we can say embryogonal mass of a division, and redivision is formed. So the wall of Vender becomes we can say two layered and forms a caliptrus. Okay, in this picture you can see it, opening of the archegonium Vender with the fertilized product embryo. So in the next slide, that is the structure of Sporophyte. So the Sporophyte of Riccia is the simplest in organization among the Bryophytes. So it takes place, we can say it does not possesses foot and the seta. Only globular capsule is present in the tissues of thallus. It is the more or less spherical, having single layered wall. The zygote produces a wall. It increases in volume and nearly fills the cavity within the Vender. So simultaneously what happens, the Vender cells divide we can say periclinally and then by anticlinal walls and enlarge in size. The Vender forms a two-layered claptra. So it encloses the developing sporophyte. A large number of spores mother cells develop within the capsule. So what happens, each spore mother cells divide by meiosis and forms four spores. So they are haploid and are arranged in a tetrahedrally. In the image you can see it. The mass of the spores enclosed within the calyptra is designated as a sporangium. So cells of the calyptrus and capsule wall gets disorganized to form nourishing fluid for the developing spores. As the capsule walls are disintegrated, the sporangium possesses no sporophytic cell. The mature spore, that is a haploid one, remain enclosed in the cavity of the thallus of the Riccia plant. So the spores in the Riccia are not liberated immediately. So they are set free after a year or so. So there is no spherical mechanism for spore dispersal. So it occurs by decay of capsule wall, calyptra, and overlapping tissues of the thallus. So the spores are carried by air or splashing raindrops. In the image you can see it, the sporophytic structure, the spore tetra formation and the distinguishing jacket in it. Now the next, that is the structure and germination of spores. A fully mature spore is uninucleated and pyramidal in shape. It has three wall layers that are easily distinguishable. So the exosporium is the outermost layer. It is highly cutinized one. Then the mesosporium is the middle layer. It has a three concentric layer. It is also cutinized, we can say. So the endosporium is the innermost layer. It is made up of pectose and the cellulose one. So the death and the decay of the thallus takes place, and the mature spores are carried by air, or we can say splashing raindrop. Spore germination begins with the rupture of the two outermost layer, exosporium and the mesosporium. The endosporium uh, protrudes in the form of germinal tube. Simultaneously, the rhizode is also emerged out. So the germ tube becomes 8-celled by division. It is called as a proteinema. It divides further and forms a new thallus. So these are the mature spores of different species of the Riccia plants. So thank you very much.